May we share? Okay, great. Well, welcome to the bi-weekly webinar. My name's Sean Brown. I'm the director of the Pittsburgh Supercomputing Center and a representative of the XSEED organization in the U.S. This is the International COVID-19 Knowledge Exchange webinar. It's a joint activity hosted by XSEED and PRACE, and I'll allow my colleague from PRACE, Matej, to introduce himself as well.
Hello, so I'm Matej Prapatnik from Ljubljana. So I work at the National Institute of Chemistry, where I head the theory department, and I'm also professor of physics at the University of Ljubljana. And I am the current chair of the PRACE Scientific Steering Committee, which is a counterpart of XSEED in Europe. And I welcome you to this webinar, which we organized together with XSEED. So.
Great, thanks, Matej. Yeah, so this activity bore out of the urgent computing activities that are going on in the U.S. and Europe, and we've expanded to all over the world. So I really welcome the new people that have joined us today. We're really happy to have you and to have your contribution. One general note that I wanted to point out, and we've been interested in understanding what open data sets are out there to share both results and primary data. I know we've had a little bit of a discussion on the Slack channel about some stuff from the Nature article that people were trying to use for genomics data, but we created a Slack a channel in our Slack workspace for people to share links and information about open data sets that they have available. And we'd really appreciate if you have any that you know of that the community might benefit from, please post them there so that we can start disseminating and getting the word out about those data sets being available. So thank you very much. So let's go ahead and get started. We'll have just like the last few times, we'll have a series of people that have volunteered to share some of the results and work that they're doing. And our first talk today will come from Gurjot Kaur. I hope I'm saying that right. I gave it a little bit of a French flourish, so I apologize if I said it wrong. She's the associate professor.
But I'm Indian.
She's associate professor at the School of Pharmaceutical Sciences at Shoolini University, and welcome and thank you for giving a presentation. You should be able to share your screen if you want to share some slides. If you don't have any, that's fine too.
Yes. No, I'll share my slides, but I just want to say that I'm calling from my phone because it's almost 10 here. Oh. And I have to kind of, be quiet because everybody is sleeping.
Oh, so thank you so much for joining us and giving us the presentation.
I'll just try. I want to share the screen, but this says host disabled attendee screen sharing.
Oh, that might be up. Okay, you should be able to do it now.
Okay. Okay. Sorry, just 1 second.
No worries.
Right.
Yep.
All right, so I'm very happy to present here, and I have a lot of queries because I'm a cellular and molecular biologist, and some parts of the projects are a little bit difficult for us. So we are more than happy if any of you can point out any information that you can help us with. On the top corner, you can see the Shoolini University. It is in the foothills of Himalayas. And therefore, one of the aims of the university is to use the knowledge about indigenous plants and the phytoconstituents and how we can use them in modern medicine. And that is how this project was born, which is called Assessment of Phytochemicals present in these plants through in silico methods for COVID-19. I took the liberty of also showing you my team. So this is me on the good days. And here is my molecular docking team and molecular dynamics team. This molecular dynamics team we are still working on because we are really newbies here. Most of them are master students working from their villages right now. So we are more than happy for the resources that we get through the consortium. At least not when we have electricity cuts. Anyways, jokes apart, we also have some advisors to the project to help us with the different parts of molecular docking and molecular dynamics. Here is the main hypothesis. Indian medicinal plants have always been known in traditional medicine to have antiviral properties. Modern research has helped us identify phytoconstants, which are have been shown to be active against known pathogens like dengue or HCV or hepatitis B. So we just aim to utilize these already known phytoconstants because of the structure similarity perhaps against SARS-CoV-2, and the first steps are in silico prediction using molecular docking. Here I'm just giving you an example of a paper that has just been published about Vidanone, which comes from Vidania somnifera, and how the authors could show that it inhibits or affects the binding between ACE2 and RBD. So we are aiming for such a study by the end of this whole consortium project. Just to kind of, for simplicity, I have written down what are the project outlines, and we are aiming at using 3 molecular targets on COVID-19, or let's say on the pathway, because human targets can also be used. And we have a list of 35 phytoconstants that have proven antiviral activity. The green just shows you what parts have been worked on, and the first one has been MPro, and we have gone through many PDBs, optimizing and everything, and then molecular docking with 35 with internal validation redockings, and we have come to 4 phytoconstants that we want to check with molecular dynamics. We are on parallel working on the literature of second molecular target, which is our human TMPR SS2 protease. So this is this was a lot of information, and I hope all of you can read this. Otherwise, I'll walk you through. I just put all the different, just for making it colorful, I put all the different dockings in the background because there were a lot of docking, and I thought maybe nobody is interested in seeing failed dockings too. What we did is we first made the list of phytoconstants and we collected articles. We analyzed really extensively at this point to really have very good scientific evidence as the basis of this project. Next, we looked at the preliminary drug likeness, if these would be available if given by inhalation. And this already, cut down some of the phytoconstants. And then we looked at the binding sites. We even assessed them other than the papers that are available with the PDBs, because we wanted to see what kind of data everybody is reporting. And you would see that even in this list, you don't see the same amino acids every time. So this, I think, was a little bit of a problem for us, but it was a good exercise. And then we also redocked the known ligand. We did preliminary molecular docking studies, and we ran like 2 parallel, 2 individuals, 2 runs. Also made a list of what kind of amino acids show up there, and then picked the best docking results. So we ended up with, like, I think this is 11 or 12 phytoconstants, and then more validation with the grid sizes, and we came up with 4. And actually, if you ask me, I only like the 2 results out of this, which we are going to take for molecular dynamic simulation. So this is where the project is, and we have a little bit of a problem, but this could also be because we are not able to judge properly. One of the PDBs was 6LU7 that we used, and unfortunately we couldn't redock it properly, and this is what I'm trying to show here. And if you see on the right side, this was the last image that we could get, and we always had the problem with the cyclic rings. We could not get a better pose than this with these specifications which we standardized. This is just to show you the binding interactions which we can we could only redo 4 out of 7 that are reported on the crystal structure. The best redocked pose came when we accidentally deleted the pentyl ring on one side, and then we had even a better fit with a binding affinity of -7, which is not bad. And these are all the different parameters that we have standardized. So if somebody could help us understand how good we are in redocking here, or what possibly we could do other than these things, maybe you have a better idea about what is wrong with our cyclic rings, that would really help us. So this is where I will stop. This is my last slide. Please let me know if you want to tell us something.
Great. Okay. Well, thank you so much. That was a great talk. Does anybody have any comments or questions that they'd like to share? You're welcome to either share them in the chat window, or you can unmute and ask as well.
I have a question. This is Jennifer Klein from Wisconsin. What docking software were you using?
Sorry, docking, Autodock Vina.
Okay, okay. Have you tried other programs like Glide to see if you can get?
We don't have access to Glide.
Okay, okay. We've found that they're pretty similar anyway, but we do get different results based on the grid size that we're using, and it sounds like you are varying that.
Yeah, we think so too. So we came to the grid size of 20 angstroms, and that's when we got this result. But we are not able to get any better, but we are not also able to tell if this is a good redocking or not. So if you can tell me something by looking at our redocking, that would be really nice.
Something pretty similar happens with ours. In Autodock, we weren't able to change the number of iterations. So it was almost as if it wasn't converging. So, you know, if we would shrink the space a little bit, then we would find it would converge more often. When we started using Glide and I could change the number of iterations, then we could nail it every time. So it seemed to have something to do with just that the amount of space that you were trying to explore and the number of iterations within that space. So you probably just need more iterations.
Okay, all right.
That's my hunch.
Oh, that sounds okay. Okay, thank you.
Or we can work together on it. You know, I can contact you.
Oh,
that would be really helpful for me too.
That would be really good.
Okay.
That would be good. Thank you so much.
Yeah, thank you. Thanks, Jennifer, and thanks for a really nice presentation. We're in the interest of time, we'll have to move on. I know we're all on the Slack here in this one, so you guys can get in communication there as well. So thank you very much for joining us so late while the house is quiet to give us a presentation. We really appreciate it. Okay, I'll turn it over to Matej to introduce our next speaker.
Yeah, the next speaker is Gatuan Sardina. I hope I pronounced this right. Assistant professor in sustainable process engineering at Chalmers University of Technology in Gothenburg.
Gaetano, are you on? Okay, well, I guess I don't see Gaetano on, so we'll have to, okay, well, we'll just move on to the next talk then. I thought
we can postpone and then.
Yeah, we'll postpone it, yeah. Something must have happened, he wasn't able to join us. So we'll move on to our next talk, which is Tom Cheatham, Director of Research Computing and the HPC and Professor of Medicinal Chemistry at the University of Utah. So thanks, Tom, for joining us, and looking forward to the talk.
Great. Hey, nice to see all my science friends here, particularly the European ones. I was supposed to go to Europe about 3 times this summer, but now we're not going at all. Do you see my slide? I can play it first.
Yep, we see your PowerPoint.
Okay, there it goes.
Yeah,
great. So we're working on 2 COVID projects here at the University of Utah. I'm only going to talk about one today, and that's trying to find peptidic inhibitors for the COVID-made protease. Rodrigo, my research professor, is doing most of the work. He's on the call today. And our experimental collaborator is Professor Eric Schmidt, who's in my department of MedChem at the University of Utah, who does experimental work. And it's kind of a workflow where we are using Rosetta to take the peptide backbone and randomize the side chains subject to energetic criteria to predict new peptides that might bind to the main protease. Then we do simulations with Amber, molecular dynamics, using the new FF20SB force field OPC water model. We were awarded an allocation from the HPC COVID-19 consortium on blue waters for 2.7 million hours. We've burned well more than that. There's a 48-hour queue on blue waters. With the K20X GPUs, we can get about 120 to 150 nanoseconds per peptide candidate. And the aim is we want about 10,000 models for each of 2 anchor positions. So with PepSec, you pick one residue is fixed, a side chain, keep it, and then you randomize the others in the 6mer. And then after you do the simulations, you use MMPBSA to estimate the free energies. We're kind of extending some of these simulations a bit longer to see if the free energy gets better. We're also looking at the initial hits were high in glycine, which makes sense because it's easy to put a glycine side chain in randomly. Larger side chain is going to be less selected. So we're filtering it so you can only have one gly. Cysteine is a problem for experiment, so we're trying to get rid of cysteine as well. And we're also doing 8mer candidates. Now, this came out of a workflow we previously developed around 2015 for Ebola, and particularly peptides targeting membrane fusion protein. But we never got to the experiments. I mean, we had an experimental lab here that could do it, but they never did it. One thing we see is there's absolutely no correlation between the randomized Rosetta peptides and what we get after simulation with Amber. So essentially when you run MD, you move to a different place. So if you look down in terms of low energy for Amber hits, it's a huge range of Rosetta estimates. And the blue and the black separation there is kind of above what the experimental binding energy is versus below what the experimental binding is. What we did see is we did find a lot of compounds. So in this black histogram, we found a lot of compounds that were better binders theoretically than the crystal structure, which is in this kind of blue line. And if we took the best hits from the first round and ran it through this cycle again, we shifted the whole curve and got even better binders. We haven't done that this time yet, but we're planning to. Now, when we first got into lockdown in the middle of March here in Utah, we started up on Longhorn Attack, which we were friendly users of. It's a new part of the Frontera machine effectively. It's got about 400 RTX 5000 NVIDIA GPUs. And we ran about 200 peptides through the workflow. And we found a couple that bound better than the crystal structure, which is where the line is here, which suggested we needed to go to more. So that's when we applied, got the blue waters allocation to get the more like 10,000. And from this distribution here, you can see when you run around 10,000 per anchor, the peptide sequences we're finding are shown above here. The anchor, in this case, is leucine at residue 3 or valine at residue 4. Again, too glycine rich, which is why we're doing the filtering now. But you can see we find a lot of compounds, a significant amount, that are better binders theoretically than we found from the original peptide. Their sequences shown here, some, whereas the canonical crystal binders are about -12 in terms of free energy, we found some even, -40 kcals. Now, the absolute value of these things is not a great indicator, but generally the trend is good. So is it really, 25 kcals or more better binding affinity? Probably not, but certainly orders of magnitude better. So it's kind of exciting. Now, the really exciting part is that the Schmidt lab cloned and expressed the protease and came up with an assay to validate these. and they bought some of these peptides a couple of weeks ago, and we just got the results within the past week. We found that our best predictor binder, this -51 is wrong, it should be 41, but the best one we bought actually is a good inhibitor. It's about 70% inhibition at 100 micromolar, IC50 about 32 micromolar. Others also were predicted to bind, but it turns out they're substrates for the protease, so they do inhibit, but they get chopped up so quick you would never use it as a drug. And in fact, you'd never use a 6-mer as a drug. What the intent is down the road is to take these, cyclize them, make them look bigger, make them more into a drug-like candidate. But for the one that does inhibit at about the 32 micromolar IC50, it's stable and it's not a substrate for at least an hour and a half so far in the tests. So we're seeing success. So we're going to continue this. We hope we're going to ask for a supplement on Blue Waters or Longhorn to continue these. But we want to do a second round refinement to see if we can shift the curve a little more to get tighter binders. And we've decided to make this data open because it is kind of very preliminary in a sense that it's just a lead compound. It's not a drug. We're not going to get IP off this or anything else. And so we'll make full trajectory data available for the top 1,000 hits in each of the different experiments we've been trying. So that's where we are. And I'll be happy to answer any questions.
Hey, Tom, I'll take the first question. Where are you guys gonna, how are you gonna make the data available? Are you gonna put it in like MolSci or something, or are you gonna just post it there?
I'm thinking MolSci if they'll do it, and they asked previously if we would do that, and I think they will. Otherwise, I've just put stuff on the web at Utah before, and that's been helpful, like our Anton trajectories of DNA and other stuff we've put there, and people have been able to access it.
Great, cool, thanks.
The internet seems to work.
Yeah, that's still, thankfully still works. Okay, anybody have any questions they'd like to ask? Please, you can ask in the chat window or unmute and ask. Any other questions?
I have a question. Tom?
Yeah, please.
Yep.
Very nice talk, thanks. So you showed results for anchor at 3 and anchor at 4. Can you combine those two anchors and put like, what was it, valine and leucine?
I believe you probably could do 2 anchors, but we don't need to. In fact, what I'd like to do, what we do in the second round of refinement is we choose the best hits. And what I would do is take the results from anchor 3, but then anchor residue 4, which is going to be different from what it was in the first round, and vice versa, so that we get more diversity, so that we don't have to necessarily anchor the same residue.
Cool.
I'd rather have more diversity than tightening the anchor.
Cool. Thank you.
And we're barely hitting any of the full peptidic space. I mean, 6 to the 21 in terms of amino acids is a huge number, or 20. But the fact that we can actually find things that are better is kind of shocking to me, with limited MD simulation and limited Rosetta exploration. So I didn't trust that the Ebola results were real until we could do the experiment. So it's really kind of amazing that we're able to show we could predict at least a couple peptides that do bind better than the inhibitor and inhibit this enzyme. So it's giving us encouragement to continue.
Yeah. Pretty cool.
Cool. Great. Any other questions for Tom or comments? Okay, great. Well, thanks, Tom. Really nice presentation. Appreciate you coming in and giving us a talk.
Sure, and hit me up on Slack if you have any questions or comments. And I'm sorry I have to drop off because we're interviewing faculty candidate at the moment. I'm missing that, so I should go back to the faculty candidate.
All right, well, thanks for joining us and giving us the update.
See you.
Yep, see you later. Then I'll turn it over to Matej to introduce our next speaker.
And the next speaker is Afshin Beheshti he is a bioinformation at NASA, adjunct assistant professor at Rogers, and visiting researcher at Broad Institute. Please, Afshin.
Yeah. Hi, how are you? Is my camera working? I look frozen on the
Yeah, I know we don't see it, but you can try to share your screen if you.
Okay, let me try sharing my screen.
Yeah.
So yes, I usually go through a VPN to log in because it's the government. So sometimes the VPN. Oh, there you go. Now.
Oh yeah, well I see me now. Yeah.
All right, I see Sean. I don't know if you see me yet.
Now we see you. Yeah, now we see you.
Okay, yeah. Sorry about that. Yeah, it's just that sometimes a VPN at NASA causes issues. Let me. Oh, did I stop sharing my screen?
Yeah.
Sorry, hold on. All right, back in action. All right, so let me go presenter mode. So yeah, thanks for the introduction. Thanks for having me present. So yeah, my name is Afshin Beheshti. As you guys heard, I'm also has a visiting research point in Broad and so on and so on. But you can, this is my email contact here. You can email me in these things, and then also some of the Slack channel and so on. So what I want to discuss today is I'm at NASA Ames. I'm a bioinformatician there, but I started kind of sort of a grassroots movement back in March. Another COVID consortium group, which we got awarded an HPC from the NASA Ames supercompute, which are part of the whole like seed cluster. So briefly, I just want to give you a little history of COVID, which is the COVID-19 International Research Team, which I lead, which anyone here can join. Sean actually joined our group and been attending some of our meetings. But you know, so others here that are interested in joining, let me know. But just a brief background, you know, at NASA I do two things. One, I have my own lab and I work on my own research. And the other, I'm part of this developing this platform called Gene Lab, which is basically where all the omics of space biology ends up. And you know, we're right now pretty much going to the next phase of development where we're working on our visualizations and things like that. So people, when they analyze their omics, you can display it on there. But here's a link here to look at that. So during that, I also lead these analysis working groups, which have scientists from all around the world come along and you know, work with Gene Lab and on various projects and, you know, work on papers to get out, things like that. And within that group, a few people were working on COVID-19 back in March. And so we decided to form a little offshoot started with like 10 of us, and this is where Covert started. And then what happened was now we have about 150 members. You can see the map distribution here of all the members from Europe, South America, and you know, the US. Unfortunately, we don't have anyone from the African continent or Asian continent, but we're more than happy to get scientists from all around, you know, globally to contribute as much as possible. And the whole idea is that we're going to have an open science platform or open science consortium where, you know, everyone's working together for a common good and we share data. Currently, we have bioinformaticians, we have clinicians, we have people who are virologists who actually are, and I'll show you some of this data in a second, where are actually doing experiments with our group where, you know, if you have targets to inhibit, they actually they will develop that and then, or they will actually test those inhibitors in their BSL-3 labs, their COVID-19 models. And then we also have people who actually can design inhibitors that we come up, targets that we come up with. So, you know, we have the whole range. And the vision, like when I set this group up, was basically, you know, to have not only people could come up with the potential drug targets through the computational methods that we all do, but also actually make it into creation. So that's what our group is. And we're pretty open with, and we have a bunch of different subgroups that people work on. We have a modeling subgroup that Sean had joined, where, you know, they were looking at different ways they could model, you know, all the data that you see for, you know, that you just see, you know, based on, let's say, population doubling of COVID-19 or the mortality rates and how can we prevent that based on, let's say, shelter in place and so on. And there's other modeling that they're trying to do based on the omics data too. And we had just a symposium in April, and we're having our second one Friday, July 17th. I just put a little plug for that. People are interested in signing up. We're almost finalizing all the speakers and put the registration up soon. I'll put that in the Slack channel also. So some of the things we've done, I mean, this is kind of a general overview, so this was really short, but we're doing a lot in this, and I'm involved in a lot of these projects too. So this is the HPC from NASA. So the NASA HPC award that we had received, we got a lot of compute time. And what we've been doing, we got RNA-seq data from, right now we have 900 positive negative patient samples sequenced through the whole RNA sequencing by Chris Mason out in Cornell. He's part of our group. So he's sharing the data of the group. And then so we're doing GATK analysis on that, running like basically the high throughput going through there and getting that data crunched out. So then once we have the high order data, we distribute to our group. And the various members of our group of access, we were giving access to HPC resource at Ames and NASA Ames, are going to do various analysis from mitochondrial analysis, which I'm going to briefly go over. That's one thing. They're going to do specific viral analysis. We have the microbial components that we're pulling out of the sequence data that we're actually looking at. So people are going to look at that aspect and the host and viral interactions. And then people are also going to be looking at different things like we have another member in our group from Georgia Tech who knows about ancestry. So he's going to be using his ancestry models on the RNA-seq data, doing some high computing to actually create and look at, if COVID-19 tends to, what biological impact does it have in different ancestral backgrounds, things like that. So, and I said, there's therapeutics that like a CEO of this company is involved. Actually, once we come up with the targets, they're going to be actually helping us out and, basically do a high throughput target inhibitors based on whoever comes up with inhibitors to come out. And I'm looking at specifically microRNAs and mitochondrial impact. And one of the microRNAs that I've already targeted, we've started testing with one of the covert members with the hamster model. We did the viral screen, and this microRNA was actually shown to inhibit really nicely the virus itself on a targeted viral screen. And then so that's why we started some hamster trials starting actually this week, early this week it started with some people at Colorado State University who's part of covert. Some of the accomplishments and papers that we're working on. This one is going to be submitted tomorrow. This paper up here. So these covert members, which I'm close with too, but Todd, he's our other co-lead. He's looking at the hidden, this is population diversity in SARS-CoV-2. So he's looking at the frequency allele differences and mutations that occur in SARS, and he's finding some really interesting novel findings here, where they've been looking at different strains from the New York population based on the data we have, but then they also dove into the public databases and started scouring they have population from the European population, they have also from the Asian population and the Chinese population. The data is available from there, and they've been finding that it's interesting novel findings about how these subtle allele differences are happening that could cause maybe different strains to be more potent than others, or maybe not, and also some biology there to show how COVID-19 might be targeting some specific pathways. Chris Mason is part of our group. This is his data. This is in bioRxiv. He's working I think he already submitted this to another journal, but in this data set, they also have environmental data. So what he did was swabbed New York subway station at the point before people went shelter in place. So it was pretty active. And so in this paper, in addition to the positive and negative COVID-19 patients, he also looked at the environmental sample. What's really interesting from that data is he showed in the New York subway, just on surfaces alone, that there's absolutely no trace of COVID-19 in the peak of people were actually going through it, walking through the subway stations, and also that's the peak when things were rising up, unfortunately, from New York. So that kind of indicates, well, doesn't last on the surface too long, possibly, and it's really because of the close vicinity of people together in that area that might be through the air transmitted. And there's these other papers here that we're probably trying to put out in a few weeks. And I'm going to specifically just talk about the mitochondrial suppression in the next slide because of some time. But there's all these other papers that we're working on, which we can discuss with people here later. So mitochondrial story. This is a really quick snapshot of it that we're probably try to submit in a couple of weeks. So basically, we early on, when we started analyzing samples from before we had this RNA-seq data done, we were analyzing bronchoalveolar lavage fluid data that was available publicly from the Chinese population from Wuhan, and we found that there, when analyzing the pathways, that we found that mitochondrial suppression is heavily regulated, by the COVID-19 in the host. So then we got the RNA-seq data from the nasal swabs, and indeed, this is what's happening. So if you look at all the distribution of the mitochondrial genes here, there's some up, there's some down, and this is with viral load. So this is viral load is determined by the PCR tests that everyone does. So the CT values and PCR. So then we see that, when there's low, medium, and high, and this is just positive and negative, viral load independent, which would make sense is closer to the medium, so it should be the average. There's a distribution of mitochondrial genes that are up, and there's distribution that are down. But when you look deeper dive into this, which is this plot which you cannot see because it's so small, is that basically the ones that are down are the really important mitochondrial genes that you want to be functioning so you have energy, fuel, and everything else. the ones that are up are the mitochondrial related genes that are basically compensating for the ones that are down. And usually, you know, they might be able to recover some of the mitochondrial functions if these important mitochondrial genes are not impaired so much, which in this case, in the high viral load, it is. So these genes that are mitochondria that are up, you know, are trying to overcompensate for everything that's happening, but in fact, you know, it's not doing it can't do the job it wants to do because the important ones are so deficient. So the idea is, you know, if you think about all the people who are hit the hardest with COVID-19, the population. So the older people, as you grow older, you got your mitochondrial functions decline. People who have diabetes or the obese are being hit harder. And those people, again, have mitochondrial functions decline. And so on, like cardiovascular disease, any populations you hear about that are hit hard, their mitochondrial functions are naturally declined because of whatever state, you know, the disease that they already have, or because of age and things like that. So the story that is coming out is pretty interesting. And then we're also tying in a lot of things that we're starting to find with glycolysis. Some other people are glycolysis experts in our group, that they're tying in that, you know, glycolysis is really high and active from this data that we're analyzing. And what's interesting is that the link between glycolysis and mitochondrial is really prominent. And we have all these experts in our group, like Doug Wallace, who's the founder of mitochondrial genetics. He's driving that aspect of it in our group. And then like Waldemar Prieb, who's a glycolysis expert at MD Anderson, he's helping out with that aspect. So we got a lot of nice synergy happening between the groups. And then also this plot here, this heat map here, just showing you the regions you can find. These are, you know, different regions of the SARS-CoV-2. There's a nice paper out there that has determined the prey genes, they call it in the paper. So they said, you know, what genes are targeted in each of these regions. So if I take these genes, overlay in the mitochondrial genes, you can find a nice distribution of what's happening with the mitochondrial per region of SARS-CoV-2. And you can see overall, again, an overall downregulation of a lot of important mitochondrial genes if you actually zoomed in and looked. And then, you know, interestingly, there's some regions that are more targeting mitochondrial than others for SARS-CoV-2. And we can get into that more, but for the sake of time, I'll move on. And then again, the certain mitochondrial pathways. And then just to end my little presentation for you, you know, just to save the date here, I can give you guys more information probably by Monday or Tuesday about the confirmed speakers. But we got a lot of really nice confirmed speakers for this. So we got people from NIAID and other institutions that I think will be of interest for people here. It's July 17th at 10 a.m. Central Time. So it's about a 6-hour symposium. People come and go as they want. And if you want more information on COVID, email me, or I should put the website name here. It's www.cov-irt.org. So, and if you want to join COVID, that's another meeting. But you know, we have a lot of interaction with people. We're more than happy to include as many people as possible. So sorry, I really quick, but
thank you. I know that was great, Afshin. Thank you very much. Yeah, and I just recently joined, so I'm excited to be a part of that. And I know Afshin is on our Slack channel, so you can also ping him there if you're interested in joining as well. Do we probably have time for one question? Does anybody have any questions or comments for Afshin?
I have a question. This is Eve. So I'm really curious what kind of metadata you have. So we've done a study about African Americans versus European Americans and gene expression in general, and we're focused on the genes associated with COVID-19. And one of the problems that we have in our analysis is we use existing data, is there's almost no socioeconomic data like zip code and things like that we could use. And there's rarely race that's given. I think you guys have race. So I was just wondering how you deal with your metadata about each individual.
Yeah, so I think that's an issue everyone has. So the metadata, yeah, that's a big issue, I think, with all the public data and the data we even have from Chris Mason from Cornell. He luckily he can control that, but his IRB, he's been going through IRB hell trying to get that metadata available to a group of like 150 people. That's in our group, as you can imagine. But next, so we've been just dealing with no metadata at this point. We've just been dealing purely looking at the viral load dependence of the information we have and doing the analysis that's independent of that. But next week, he's actually going to be able to release some of the metadata to our group. So there's going to be the data that you're wanting to have.
Yeah.
Going to have age and sex, and I think what else did he say? I think we should have also racial profile or ancestry, so we know exactly what you're trying to get at. Like then we could actually then categorize the data, for people who are different socioeconomic situations based on their ancestral profile or their race, you know, so hopefully, so I mean, if you wanted to join the group, let me know, and you know.
Okay, I will. I tried once and it didn't happen, and so I think it was a glitch.
Oh.
So I'm gonna
Yeah,
get back to you.
there might have been a chance that we might have missed the email sometimes. So sorry about that.
I'm real excited. Thank you so much for doing what you're doing.
Yeah, no. And as I said, anyone here, if we don't happen to reply back to you within a week, that means we somehow missed your email, and keep harassing us until we reply. So.
Well, thank you, Afshin. That was great. And good luck with the consortium. I can't wait to continue to be a part of it. So let's move on to our next speaker will be André Cisneros. I think I said it right. We're professor of chemistry at the University of North Texas, not Northern Texas, North Texas. So please, thank you for joining us and giving us an update.
Thank you. Thanks for the opportunity. Okay, so can everyone see the PowerPoint?
Yep.
Okay, awesome. So we're working on QMM and molecular dynamics using polarizable force fields to investigate the reaction mechanism of the main protease and RNA-directed RNA polymerase. And so before we start, just want to acknowledge that my group and I are on Wichita land and Black Lives Matter. So there are several grad students and postdocs that are working on this. The results that I'm going to show you today are basically a snapshot of what we've been doing. We're also collaborating with Jean-Philippe at Sorbonne and Peng Yu at UT Austin. We're part of the Tinker developer team, Tinker developers, and also collaborate with Amber developers. So we're really excited that we got time from Exceed on Stampede and Summit and Azure. And we also have a little bit of time on Longhorn. So we've been working on some of that. Like I said, the goals are to investigate the reaction mechanism of different inhibitors on MPro and RDRP using Amoeba MD and QMM with Amoeba. So the very long MD with Amoeba is done in collaboration with Jean-Philippe, and we're basically looking at the reaction mechanism. For that, we're using our code, which is called LIChem, Layered Interacting Chemical Models. And it's basically a wrapper code that allows combining different quantum and classical codes. So we have capability for Gaussian, NWChem, Psi4, and then Tinker, LAMPS, etc. And we allow for explicit use of polarizable potential. So we have a better description of not only the quantum part, but also the environment. We developed corrections for long-range electrostatics in QMMM, which are in those papers. And we also have a hybrid parallel reaction path optimizer called quadratic string method, which is the algorithm that we're using for the path optimizations. So I'm going to tell you some progress that we've made. This is mostly MD results, both for the main protease and RDRP. So it's a C3-like cysteine protease. So what we decided to do was take some of the structures from Diamond in the XChem fragment screen, and we're focusing on 3 inhibitors from this COVID-19 summary. So PCM 1026, 10, 35, and 835. They were docked to 6 LUJ, and we're also taking the alpha ketoamide peptidomimetic in 6Y7M, and we're looking at the reaction mechanism. So right now we've run some amoeba dynamics on the PCMs and Amber and Amoeba on the alpha ketoamide, and just looking at the reactive distances from the cysteine to the carbon that's going to suffer the nucleophilic attack, and the activation of the cysteine, so pulling the hydrogen so we can have the nucleophilic attack. Some of these inhibitors are not going to be reactive. So for example, this alpha ketoamide and this PCM, these are preliminary results, but that's what seems to be happening. But one of these PCM 102635, we call them A and B because they have chiral center, so they're chirally active. These seem to be held in place in the active site, and so we're going to be testing these for QMMM simulations. So we've started with QMMM optimizations. I know that we're running short on time, so I'm just going to skip this. And this is just showing a linear interpolation for the reaction mechanism that we're going to be testing. So proton transfer to the histidine, and then nucleophilic attack of the sulfur to the alpha carbon, and then followed by proton transfer to the beta carbon. This is basically a thioene mechanism that we're exploring, and hopefully we'll be able to tell you some more progress soon. With respect to RDRP, it's a multi-subunit polymerase that has possible proofreading activity. We're setting up a collaboration with people in Mexico and in Galveston for this phospholyxnuclease. Hopefully we'll get some structure soon. So when we started, there were no ternary structures of RDRP with template and primer RNA and incoming triphosphate. So Ser Nasimikan in the group took polio and norovirus homologs and developed our model with incoming both RNA triphosphate, DNA triphosphate, remdesivir, and a remdesivir analog that has this code in PubChem. We've done triphosphate and templating base parameters, both for Amber and Amoeba. We have to share those, and we'll upload them, upload everything to MolC. And we've run 50 nanoseconds of initial Amber MD for several different systems. So just to tell you a little bit about the initial structure, the mechanism of RDRP is basically copying the viral RNA. And in order to do that, it needs incoming ribonucleotide triphosphates. And so what Sarah did was, using our knowledge from DNA polymerases, she generated the initial structure from RDRP. And very recently, last month, the structure with remdesivir and the templating and primer bases was reported. And so when we overlaid them and calculated the RMSD, we're at less than around 1 angstrom, less than a lot of the places for the entire protein. And in the active site, we decided to include 2 metal ions, in this case magnesium, because those are the ones that are going to support catalysis. And so right now, what we've been doing is setting up several different systems with different incoming triphosphates, including RNA triphosphate, DNA triphosphate, remdesivir, and this CID, I'm just going to call it CID, in the incoming nucleotide position. But from other papers in structural biology, it's also known, well, it's been proposed that remdesivir actually doesn't stop the polymerization at the incoming nucleotide point, but further down the line. So we've decided to investigate several other different systems, including remdesivir, at different positions further downstream from the primer base. So the primer base is this A naught, and A1 is the incoming nucleotide base. And so for the incoming nucleotide, so far we have these 50 nanoseconds of MD. The RMSD is looking okay, so the structure is not falling apart, which is great. What I think is really interesting is that the reaction mechanism is going to involve activating the oxygen in the primer base. So a proton has to be abstracted. There are several different possibilities for this. One possibility would be to have this aspartate activate that oxygen. And the next step is a nucleophilic attack on the O3 prime on the alpha phosphate. So in order for that reaction to happen, we have to have the oxygen and phosphorus resistance relatively close. Interestingly, when we have RNTP, in this case it's ATP, the because of the complementary strand, we see that starting from the MD, the incoming nucleotide actually gets a little bit closer, which is really great, and then it stays nice and tight. Other systems which I think are interesting, and these are again all preliminary results. For example, with CID, which is, like I said, an remdesivir analog, but instead of having a cyano at C1 prime, it has 2 methyls at C1 prime and C2 prime. In this case, after MD, the triphosphate really just does not want to be there, and we're running more dynamics to confirm this. If we have remdesivir, the results are not as great. It's not getting as tight as we had for the incoming RNTP. So we're running replicates on these, and when we have remdesivir both at the minus A3 position, the templating base, and the incoming base, the remdesivir does look like it's going to be staying around, and so we're going to be running the QMM simulations on there. And like I said, we're optimizing a lot of these things, and that's pretty much where we are.
Great, thank you, Andres. Any questions? We probably have time for one question, but we definitely want to. We have one more talk to get to today. Anybody have a quick comment or question they'd like to ask? Okay, great. Well, we'll just go ahead and move on to the next talk. Thank you very much. That was really great. Thanks for contributing. And I'll turn it over to Mate for our last speaker.
Well, the last speaker of today is Som Dutta, Assistant Professor of Engineering at Utah State University.
Hey, everybody. Thanks for this opportunity. I'm going to share my screen, and hopefully I'm able to show something interesting. What I'm going to talk about today is moving away from, in general, theme of talks I have seen over the last few weeks in this presentation. So I'm a fluid dynamicist. So essentially, I do large-scale computational fluid dynamics. And essentially, the work I'm going to show is how aerosols, so somebody talked about airborne transmission. So what I'm trying to look into is how airborne transmission happens in a really detailed way, and then try to come up with mitigation strategies. So we have been awarded around 1.2 million node hours on Frontera and Blue Waters. I think we will need more, but hopefully let's see how far we can reach with the current allocation. And on this work, I started as assistant professor last year, so I don't have any trained graduate student who can quickly do this work. So I am basically still doing all the simulations like a postdoc or a PhD student would do, with some help from my colleague from Lawrence Liverpool National Lab, Cheatham III. And so basically what happens is it's already known that we generate aerosols not only when we cough or sneeze, but also while we are talking or singing or even sometimes breathing. And these aerosol clouds, when they are generated, they combined with the background turbulence in the air. What is known as it could actually keep these aerosols, depending on their size, for a long time in the air column. And then essentially they could go on to infect somebody. There was a study, one of the first papers which came out, which showed that these aerosols containing COVID-19 could remain infectious till about almost 3 hours after they have been released. So now talking about the size of aerosols, so there's a little graph I have made. So if you see in general, when we speak, most of the aerosols or breathe, most of the aerosols are of the size 5 micron or smaller. And these are so small that even without background turbulence, they can remain in suspension up to almost 20 minutes or even higher. So now think of the background turbulence generated by the air conditioning system. So once we have that air conditioning system on, that these things could remain in suspension even further. So the aerosol sizes of interest for us is from around 2 micron, so around 1 micron to around 20 microns, because bigger than 20 microns, they essentially very quickly fall to the ground and they take almost less than 4 minutes. So bigger droplets is not the aim of our study. So on the right-hand side, this is basically animation from our first simulation. It's essentially showing an isosurface of the region in a room, which essentially has velocity higher than a certain extent. That is what we think is required to keep things in suspension. But then we went on to add the aerosol. So this first setup we have been running is an empty room of size 2.2 meter by 3.5 meter by 4.2 meter. So it's kind of a real size room. And there's an inlet and an outlet, and we are releasing aerosols at 4 different sources. So currently we are running a case which has around 500,000 aerosols in total. The computational size is around 30 million computational points. And I will tell you a little bit about how long these things take. What we are using is, for the aerosols, we are using a Lagrangian particle tracking. At this point of time, it's a simple model in which the size of the aerosols are not changing, but in the future, we want to also change the size of the aerosol, because depending on the background temperature and humidity, aerosol size also changes with time. And the Reynolds number of these simulations is 12 to 13,000, which is essentially equivalent to ACH, 6 ACH. 6 ACH is, which for this, for a room means 6 air changes per hour. So usually we will focus between 6 to 10. That's what hospital rooms are. In office rooms, it's even lower, and that's why I think it's even more substantial that those are more problematic. So let me go to a little bit more interesting thing to show, which would be the animations actually. So can you see my screen, the animation? Okay, so what we are seeing here is that we have aerosols, the 4 different color aerosols are being injected at 4 different points in the room. And they are being colored according to the initial point they are being injected at. And what we are seeing is, depending on, and they are the same sizes, they are all 5 micron sizes, depending on where they are released in the room, just on the basis of that, they have a very different behavior. In the sense, the resident time of how much time they remain in the room is very different. I have some other animations, but what I will do is, because we don't have much time, so I will shift back to the result. And I will post these online so that if somebody wants to take a look, they can take a further look at it. So what we found was that, just going back, so these simulate, so what if we are to simulate 1.5 minutes of real time, it takes around 768 node hours on Frontera. And so basically, so to get something realistic, we need to go around 1 to 60 minutes. That takes around 31,000 node hours on Frontera. And so we are basically running on 128 nodes. We can go on higher nodes. NEC 5000 scales very well. So, but then what is the interesting bit is, what we are trying to be as accurate as possible. So what we are doing is, we have an initial time when we do not put in the aerosols. We allow the room to reach a turbulent, statistically steady state that itself takes around 50,000 node hours. So the simulation I'm showing, the total thing will take around 100,000 node hours. And now once we go to bigger rooms or bigger settings, these will go on to be taking around 300 to 400,000 node hours. The interesting thing, what we have been seeing is from the initial results is, if you see the right-hand plot is kind of the percentage of aerosols from each size which have gone out of the vent. So what we are seeing is that depending on where you talk or where you sneeze or cough, actually the amount of aerosols that go out of the room is very different. So the hope is that we understood, people in the HVAC community have been studying these things for a while, but the tools they have used do not capture all the physics. What we are doing is, we are doing these at such a high resolution that is similar to a resolution what people in like the turbulence community we usually do. So we are bringing that knowledge base to this and to the transport processes, so that we can understand this in a more scientific way, so that we can build future mitigation efforts.
Can I ask a question?
Yeah.
Yeah. So why you said that you will go to larger rooms, but couldn't you just use some reduced dimensionless units and rescale the problem?
Right. So one of the reasons to go to a slightly larger room is it will also require. So the dimensions I have now these are the initial dimensions we thought, because this is based on some literature we saw, but we then talked to more folks. The thing with larger rooms or adding more complexities is it will generate dead zones, which are very different from what you will have in a smaller room. Because at the end of the day, there are so many variables involved. So you can even change the position of the inlet and the outlet vent, and that will itself change the way the flow kind of circulates within the room and goes out. So the reason to do larger room is it will allow us to do also larger ACH, that is air changes per hour. So that's kind of, we just want to see that the effect is very similar, just to be sure that just changing the size of the room doesn't have an effect. Because at the end of the day, there is no limits to how big a size of the room could be, right? People are in
That's why I
commercial.
was asking, if you put the reduced units, if you use dimensionless units, you can rescale everything, no?
Yeah, so we are actually using dimensionless units, but even within that, what we want to see is, even if we look in a like we just increase or decrease the Reynolds number, and we go, that basically accounts for the flow velocity, but that won't account for the length scales within the room.
Okay,
So there are certain length scales in which these secondary circulations are happening, which won't be captured, we think.
Sure. Thank you.
Questions?
Oh yeah, thanks. Are there any other questions, comments? I know we're a little bit over time, but thank you for that's really interesting talk. Something we certainly haven't seen before. So at this meeting, so that's great to get a new area that people are working on. So that's awesome. Thank you. Any other questions or comments before we adjourn? Nope. Okay. Well, thanks again, Som, for a great talk, and all of our speakers for really excellent updates and presentations. Great to see a lot of work going on out there. Just a reminder, we have a Slack channel and an email list. I put it back in the Zoom channel, chat channel there, so you can see it in case you want to join. And we'll be having our next webinar on July 2nd, Thursday, July 2nd at the same time, same channel. So we hope to see all of you there. And please, if you have anybody that you think would be interested in joining us, feel free to share the invite with them and the Slack channel and the email list, and we'll be happy to have them and give presentations. Thank you very much. Appreciate you, everybody joining.
Bye.
Thank you. Bye.
Thanks. Bye.